Today we are diving deep into one of the most incredible processes in all of biology, the cell cycle. And look, this isn't just about a cell splitting in two. It's more like a molecular ballet, where every single step is timed and executed with breathtaking precision. Getting this right as well, it's everything. It's the difference between healthy growth and diseases like cancer. So if you're studying for an exam and you need all the details, you are in the right place. Let's get into it.
So, here's our game plan. We'll start with the basic life story of a cell, its timeline. Then, we'll meet the molecular engine that's actually driving everything forward. After that, we'll navigate the crucial checkpoints that make sure everything is perfect, and witness the high-stakes drama of division itself. And of course, we'll have to look at what happens when this beautiful system breaks down, and finally, we'll check out the ultimate failsafe a cell has to protect the organism.
All right, first things first, let's get a bird's eye view of a cell's life. The whole thing really boils down to two major stages: a really long preparation phase, and then a much shorter, super dramatic phase where the actual division happens. It's a cycle of growth, duplication, and separation, and it's repeated over and over with unbelievable accuracy.
Now, for a typical human cell, this whole cycle takes about a day, around 24 hours. The vast majority of that time, as you can see, is spent in G1, or gap 1. That's where the cell is just growing and making all the proteins and other stuff it needs. Then it hits the S phase for synthesis, where it knuckles down for the huge task of copying its entire genome. After another quick growth spurt in G2 to make final preparations, it enters the M phase, mitosis, the grand finale where it finally splits.
So, you can really think of the cell cycle as a play in two acts. Act 1 is called interphase, and it covers G1, S, and G2. This is the long part, right? And for good reason. The cell is doing some incredibly heavy lifting. It's growing, it's making all its parts, and it's meticulously copying billions of DNA base pairs. Everything has to be perfect. Then you have Act 2, the mitotic, or M phase. That's the main event, where all that careful preparation pays off, and you get two brand new daughter cells.
So, what's actually pushing the cell from one phase to the next? It's not just some simple timer ticking down. Nope, it's a beautifully complex molecular engine, and the core of this engine comes down to two kinds of proteins working in perfect harmony: CDKs and cyclins.
Okay, let's meet the first part of our engine, the cyclin-dependent kinases, or CDKs. You can think of these as the powerful workhorses. They're kinases, which means their job is to add phosphate groups to other proteins, a process called phosphorylation. And phosphorylation is like a molecular on-off switch. Now, CDKs are pretty much always hanging around in the cell, but here's the catch. On their own, they're completely inactive. They're like engines, just sitting there waiting for a key.
And here are the keys, the cyclins. These are the regulatory partners that CDKs absolutely depend on. Unlike the CDKs, which are always there, cyclin levels go up and down in these beautiful, predictable ways. They get made at very specific times, build up, and then are destroyed just as quickly. It's this precise rise and fall that determines which CDK engine gets turned on, and exactly when it happens, pushing the cell forward with perfect timing.
And this is where you see the incredible layers of control. It's not enough to just put the key in the ignition. For a CDK to fire up, three things have to happen. First, the right cyclin has to bind to it. Second, another enzyme, an activating kinase called CAK, has to come along and add a phosphate group, kind of like giving it a final thumbs up. And third, you have to make sure there are no CDK inhibitor proteins, or CKIs, latched on, acting like a parking brake. It's a triple lock system to make sure the engine only starts at the perfect moment.
So now that we know about the CDK cyclin engine, let's see it in action. A cell doesn't just barrel through interphase. It has to pass these critical checkpoints. These are crucial decision points to make sure everything is absolutely ready for the next step. And these checkpoints are where that engine is most carefully controlled.
The first and maybe the most important checkpoint of them all is the G1 restriction point. This is it. This is the moment a cell fully commits to dividing. Once it passes this point, there is no turning back. It's an incredible cascade. It starts when the cyclin D CDK4 complex adds a phosphate to a gatekeeper protein called PRB. This forces PRB to let go of another protein it was holding onto, called E2F. Now, E2F is a transcription factor. Its job is to turn on genes. Once it's free, it races off and switches on all the genes needed for DNA replication, including another key player, cyclin E. The new cyclin E CDK2 complex then shoves the cell past that point of no return and straight into S phase. Once the cell is in S phase, its number one priority is copying its DNA. Perfectly. The S phase checkpoint, which is run by the cyclin A/CDK2 complex, basically acts like a quality control supervisor. It's constantly monitoring replication, making sure the entire genome is copied exactly once and without any errors. If it spots a mistake, bam, it hits the brakes on the whole cycle, giving the cell's repair machinery time to fix the problem before things move on.
Okay, so the cell has pulled it off. It's doubled its genetic material. It's now holding 2 complete sets of instructions. But how in the world does it manage the incredible mechanical challenge of separating those instructions with absolute precision and then dividing itself in 2? Well, this is where the real drama of the cell cycle begins.
And that brings us to the main event, mitosis. This isn't just one simple step. It's a high-stakes, high-wire act of precise choreography. It runs through these distinct stages. You probably remember them: prophase, metaphase, anaphase, telophase, all to make absolutely sure that each new daughter cell gets one perfect set of chromosomes. It is, without a doubt, the most visually spectacular part of a cell's life.
The master conductor of this whole mitotic orchestra is a complex called MPF, or mitosis promoting factor. And if you look closely, you'll see it's our old friend CDK1, paired up with its special mitotic partner, cyclin B. When MPF gets activated, it's like a master switch gets flipped. It triggers this cascade that causes the chromosomes to condense and the nuclear envelope to break down, launching the cell into the whirlwind of mitosis.
Now, the cell has a really brilliant way of timing this. As the cell is cruising through G2, cyclin B levels are steadily rising, forming lots and lots of inactive MPF complexes. At the same time, a kinase called Wee1 is going around and putting an inhibitory phosphate, basically a brake, on every single one. This allows the cell to build up a huge stockpile of MPF that's ready to go. Then, at just the right moment, another enzyme, a phosphatase called CDC25, swoops in, removes all the brakes at once, and unleashes this massive wave of active MPF that shoves the cell decisively into mitosis.
One of MPF's most critical jobs is to set up the separation of the chromosomes. But first, another checkpoint, the spindle assembly checkpoint, has to give the all-clear, confirming that every chromosome is properly attached to the mitotic spindle. Only then does MPF activate a protein complex called the APC. Think of the APC as a demolition crew. Its first target is a protein called securin, which it tags for immediate destruction. See, securin's whole job is to keep another enzyme, separase, in check. So the moment securin is gone, separase is free. And separase acts like a pair of molecular scissors, going along and cutting the cohesin proteins that have been gluing the sister chromatids together. The instant that glue is cut, the chromatids are pulled apart to opposite ends of the cell, and anaphase has begun.
So we've just walked through how incredibly precise this whole system is. But what happens when these carefully calibrated controls fail? What happens when checkpoints are ignored and a cell starts dividing completely out of control? Well, the result is one of our most feared diseases, cancer.
When cell division goes haywire, it creates a tumor. But it's important to note that not all tumors are the same. A benign tumor is a mass of cells that grows, but it's self-contained. It doesn't spread. A malignant tumor, on the other hand, that's cancer. It's far more dangerous. Its cells have accumulated mutations that have broken the cell cycle controls. These cells invade neighboring tissues, and they can even break off and travel through the bloodstream to start new tumors elsewhere. That process is called metastasis.
So what stops this from happening all the time? Well, one of the cell's most vital defenses is a protein called P53. It's what we call a tumor suppressor. It's so important it's been nicknamed the guardian of the genome. When P53 detects DNA damage, it slams on the brakes, pausing the cell cycle. This gives the cell a chance to repair the damage. But if the damage is just too severe to be fixed, P53 makes the ultimate call. It triggers the cell's self-destruct program, apoptosis, ensuring that a dangerously damaged cell can never go on to become cancerous.
You know, understanding the cell cycle in this detail gives us some really powerful ways to fight back against cancer. Since cancer is all about rapid division, mitosis is its greatest vulnerability. So anti-mitotic drugs like vincristine and paclitaxel work by messing with the microtubules, the little protein filaments that build the spindle needed to pull chromosomes apart. Newer drugs can even target the motor proteins that do the pulling. The challenge, of course, is that these drugs also hit our normal, healthy dividing cells, like in our hair or our gut, which is what leads to those tough side effects.
Okay, we've mentioned apoptosis a couple of times now, especially as that final command from P53. This process of programmed cell death is so fundamental it really deserves its own moment in the spotlight.
When a cell is too damaged to be fixed, or it's infected, or maybe it's just no longer needed, it has one last elegant trick up its sleeve.
You see, cell death isn't all the same. There's necrosis, which is a messy, traumatic death. It's what happens from an injury. The cell swells up, bursts, and spills its guts everywhere, causing a big inflammatory mess. Apoptosis is the complete opposite. It's a quiet, clean, orderly disassembly. It's actually essential for normal development. It's how we got rid of the webbing between our fingers and toes in the womb. The cell just shrinks down, packages itself into neat little bundles, and gets tidied up by its neighbors. No fuss, no mess.
One way this gets triggered is through the intrinsic pathway, which responds to damage from inside the cell. When the cell senses that things are beyond repair, a protein called Bax starts poking holes in the mitochondria. This allows a critical molecule, cytochrome C, to leak out. Once cytochrome C is in the cytoplasm, it helps build a structure called an apoptosome. This complex then kicks off a chain reaction, activating a whole family of executioner enzymes called caspases. And these caspases are the demolition crew. They systematically chop up the cell's proteins and DNA, carrying out the death sentence with absolute precision.
And that brings us to one last fascinating idea. Most of our cells can't divide forever. There's a built-in countdown, a limit of about 50 divisions called the Hayflick limit. This biological clock is tied to the very ends of our chromosomes, which are called telomeres. They get a little bit shorter every single time the cell divides. When they get too short, the cell just stops. This is a crucial anti-cancer mechanism, but it's also deeply linked to aging. And here's the twist. Cancer cells often find a way to cheat this clock. They turn on an enzyme that rebuilds their telomeres, essentially making them immortal. It really makes you think about this profound balance our biology has struck, a finite cellular lifespan that protects the whole organism, but also ensures that nothing, not even our cells, can last forever.